Right. So the first thing is to just maybe make a distinction between what I'll call genomic prediction and genomic selection. The difference in my mind is that the prediction part of it is focused on sort of the statistical theory and the computational practice of making these predictions, which turns out to be quite similar across species, plants, animals, and humans. And so there's a lot of sharing of knowledge that can occur between these different researchers. The kinds of predictions that one can make, there are, I think, basically three kinds of predictions: additive values, genotypic values, and hybrid predictions. So plant breeders are unique in that they care about all three kinds, whereas for animal breeders, for example, primarily are interested in just predicting the additive or the breeding value. Humans are primarily, geneticists would be primarily predicting the genotypic value. And again, plants, we deal with all of those things. So selection then is basically how do you use these predictions in breeding? And this is where it becomes more difficult to generalize, because the design of a breeding program tends to involve a lot more complexity and details related to the biology, the logistics of breeding, how do you allocate resources. So it's harder to say something about potato that maybe is as meaningful in rose or in turf grass. And ploidy is not going to be per se a very unifying theme when it comes to talking about the design of a breeding program compared to some other things about, like, clonal propagation, for example. Okay, so having said that, let's just go over some basic concepts from quantitative genetics that are relevant to understanding genomic prediction. And I think this will be review for many of you, but just so we're on the same page. The genotypic value of a plant variety is the average phenotypic value over some population of environments. And when I use the word environment, typically in plant breeding, we're talking about a combination of a location, a year, and a management system. And there are other definitions, but this is just the most common way people think about it. Genotypic value is synonymous in my mind with clonal value, because what you're talking about is a measurement over some clonal replication of a genotype. And that clonal replication could be by sexual means or asexual. This is not a common way when people say clonal, they usually mean vegetative propagation, but propagating a pure line, by seed sexually is also clonally replicating it. And taking two inbreds and producing an F1 hybrid and making the same F1 hybrid and planting it over a wide set of environments is also a type of clonal replication. So anyways, genotypic value can be relevant for all these different situations. The basic linear model is shown here. It's a decomposition of the phenotype into the genotypic value, some environment effect, an interaction, and a residual. And we can quickly then go to talking about partitioning of the genotypic value into its breeding value, and then some residual that's not part of the breeding value. The difference is that the genotypic value is the best predictor of the clone performance, but not necessarily the progeny of that clone. That's what we mean by breeding value. And one of the fundamental discoveries, in theoretical quantitative genetics is that you can express the breeding value in terms of an additive value under random mating, which is defined based on the regression of the genotypic value on allele dosage at the QTL. So the genotypic value has this additive value and non-additive value, and the additive value component in this equation on the right for a set of biallelic QTL could be written as the sum over the product of the dosage of the allele, say the alternate allele at QTL1 times this additive effect for that allele, and then adding those all up. So that's what this summation notation on the right-hand side is indicating. And now this is more than just a theoretical concept. You can find examples of an additive relationship everywhere in plant breeding. Here's one from potato, which shows for our trait of vine maturity, important measure of maturity in potato, that from one of the SNPs on our array, which happens to have a very high LD with the major gene that affects this, the more copies of the alternate allele of the SNP, we can see a stepwise decrease, proportionate decrease in the phenotype, right? This is exactly what's meant by an additive effect. Okay, so now moving from a regression on QTL, but in practice, we don't know the QTL, so we're stuck with doing these regressions on the markers. And this actually introduces a lot of nuanced complexity, which I'm not going to go into, and just kind of talk a little bit at a high level about how you transfer some of these concepts over to marker regression. The main problem being that we're going to be using typically many more markers than we actually have observations. So we can't use the methods of ordinary linear regression. One of the simplest but yet effective ways of dealing with this is to treat your marker effects as random variables or random predictors. And then we can use the method that's called BLUP to actually still get a reasonable solution. So the best predictor or BLUP of the additive value can be expressed as the sum of the marker dosages times the best predictors of their corresponding additive effects. Now, BLUP is a special case of a more general type of statistical methodology for these types of regression problems called ridge regression, and hence you'll often find the method referred to as RR-BLUP in the literature, only in the context when you're talking about specifying using the marker effects as the random effects in your variable. That's when you talk about RR-BLUP. Now, the RR-BLUP formalism has a close relationship with what's called G-BLUP, and the difference is that instead of treating the markers as the unknown predictors, instead of treating the marker effects as the regression coefficients, we're going to instead try to use the additive values themselves as random effects. And in doing so, we have to have a different assumption. The marker effects are typically modeled as being independent random effects, whereas if you have additive values as your random effects, now you have to specify the covariance between those effects. So it becomes a multivariate normal distribution. The symbol G is used to denote the genomic relationship matrix that expresses those covariances. And in order to relate the G matrix to the markers, we have to go through a little bit of some formula here. So that's what I'm trying to do on this slide. On the left, we're expressing the additive value, again, as a sum over some allele dosages times a set of marker effects. And if we take all of the additive values for all the individuals in the population, and we stack them on top of each other as a single column vector, that becomes equal to this matrix of dosages where the rows are the individuals and the columns are the markers. And then that's multiplied by the vector of marker effects. So what this matrix equation is often written succinctly just as this, this bold font expression on the right, where a bold font usually represents either a vector or a matrix, and the capital letter is conventional to mean a matrix, and the lowercase is a vector. So the vector U is equal to the matrix W times this vector alpha. The variance covariance matrix of the vector U, which is, again, the additive values, by applying some simple formulas from linear algebra, can be expressed as the W matrix times its transpose. And then you still have this variance parameter, which describes the variance of the marker effects. And this is basically the definition of the G matrix. The G matrix is defined as the variance covariance matrix of the additive values relative to some variance parameter. And there's only one unique way of defining the relationship between those two parameters to come up with a sensible way of making this work. And that's what you see here on the right-hand side. So this is the basic formula for how you compute these genomic relationship matrices from your matrix of markers. It's such a simple calculation, actually. Just as an aside, people often use software packages to do this calculation, but when in reality, it's like one line of R code or 2 lines of R code. So when you have missing data or the complexities, yes, but really this is something that anyone who knows the basics of R can do quite easily. The normalization factor that you see in the denominator here actually does depend on the ploidy. This is, I think, the only time I'm actually going to talk about ploidy right in this talk. So and that's one of the, I think, worthwhile things to take away from this is that the whole theory and mechanics of genomic prediction doesn't depend much on ploidy. It enters the process in very few places. And for the most part, this is the same lecture I would give to a group of students, graduate students in a graduate plant breeding class for regardless of what ploy they're working on.
Cat.
Okay, so a bit of a word of warning: if you have never seen what the actual numbers look like when you compute these G matrix coefficients, they don't quite look like what you might expect if you've ever looked at the pedigree relationship coefficient. So the letter A is traditionally used to refer to the relationship matrix based on pedigrees. In that case, the coefficients are based on calculating probabilities of identity by descent. So they're probabilities. They can't be negative numbers. Whereas G matrix is fundamentally not based on IBD. It's actually based on using a correlation as a measure of similarity, and therefore you get positive and negative numbers. However, they clearly have a relationship to each other. This is a real data set for 570 potatoes. You can see that in general, the higher the G matrix, it's also the higher the value in the A matrix, and you can make regression lines that relate these two. But you still have quite a lot of variation. For a given value of A, you have a lot of variation around that, which is largely due to two phenomena. One is that the G matrix is accounting for Mendelian sampling. So you expect some variation around just the average that you would get from a pedigree calculation. But it also somehow wraps information about LD, or linkage disequilibrium, in your founders. It's baked into it. And that's part of what makes it difficult to, historically has been difficult for people to rationalize why prediction works sometimes and not others, or what the trends are, because there are a lot of different things that are baked into the G matrix. And I'm going to try and unpack some of that for you here today. So one, I guess, important theoretical result is that these two different methods of making the predictions that I've mentioned, the RR-BLUP, which is where you are estimating predicting marker effects, or G-BLUP, they're mathematically equivalent if you construct the scaling in the right way. However, and there are benefits there are some practical benefits to predicting marker effects, to save them and be able to use them in other situations. But my experience, having done this for 10 years now, is that I think you're better off thinking of the predictions in the G-BLUP framework, because it more accurately reflects, I think, the real nature of what you're doing. And this again, the reason I say this in part is because when you think about marker effects, you're led to think about something which is a very local phenomenon, that the marker effect is representing, let's say, the QTL information near that marker. But in reality, there's additional information that's present in each one of those marker effects, and that is a relationship information. So a series of very clever papers, two clever papers by David Habier and co-authors, showed how both LD between markers and QTL, but also relationship, are fundamentally present in the G-BLUP or the RR-BLUP method, but it becomes clearer to think about it when you think about it as a G-BLUP, I think. And what these two kinds of information actually then have different implications for how genomic prediction works, or when it works. And I'll come back to that in a second, but I first wanted to just make sure some terminology I'm going to start using, which I want to make sure everyone is aware of. When we talk about training populations in genomic prediction, we're referring to the sets of genotypes or clones and the environments together to make these predictions. As an aside, people sometimes think that they're leery to get started if you're running a breeding program, because you think you have to do all this, you have to make a bunch of additional investment to generate these training populations. And as I'll show a bit later, that's not true. You can really just use your breeding program to construct the training population, and there are very good reasons for doing so. So hopefully, if anyone was still thinking that there was a lot of extra work required, I can dispel that myth for you right now. The validation population refers to the group that you are predicting, and depending upon, if that usually implies you already know the values, you've already measured the phenotypes for those individuals. If you haven't measured them already, you probably would just call them selection candidates, which is like in the real world application. But either way, I'll use the VP abbreviation for short, just because it simplifies some of the language. And then finally, prediction accuracy would be the correlation between your predicted and your true values. Again, one common misconception I find is that the validation population or the selection candidates is different than the training population, and that doesn't have to be the case. In fact, a very important strategy is to choose your selection candidates from within the training population. That's actually how I got started with genomic selection, as I'll tell you in a minute. And in that case, we tend to, one way we can differentiate this flavor of genomic selection is to call it genome-wide marker-assisted selection. The reason being that traditionally, the traditional marker-assisted selection implied that you had phenotypes and marker data, although it was used, in practice, it's often used, again, in more broadly, regardless of whether you had the phenotypes, but that was the original pure meaning of that term. So to try and, again, make the connection with that, I'll sometimes call this genome-wide mass to refer to a case where I'm trying to also use my training population as my selection candidates. And the practical reason you would do this is that having some phenotypic data for selection candidates improves the accuracy of genomic prediction. So especially as a conservative way to get going, it can be a good option. How do you estimate prediction accuracy? Well, there's two main ways. One is based on statistical theory. So BLUPs have a certain predicted accuracy, which is more commonly reported as the squared accuracy, and it's called reliability. The other way that's, the number one method is what you'll find often in animal breeding papers and increasingly in plant breeding papers, but traditionally it was really a method in animal breeding. Plant breeders historically used method number two, which is where they did a cross-validation. They would mask the observed values for some set of individuals and then take the correlation between the prediction and those observed values. Now, both methods have strengths and weaknesses. So I often like to do both methods and see what they tell me. The advantage or let's say the disadvantage of number one, the theoretical model, is that it assumes the model is correct. And we know that your model is not correct, right? Models are just useful. They're never really purely correct. You don't know the real genomic relationship matrix. All you can do is estimate it. So the cross-validation doesn't rely on any of those assumptions. On the other hand, to use cross-validation, you're limited by how good your observed data are. You know, unless you've measured the genotypic value or the breeding value over a lot of environments, you probably don't have a perfect estimate of the true value, and therefore you have to then try and adjust this correlation for that uncertainty, and those adjustments are not perfect either. So again, I think I encourage people to try and do both if you can. Coming back now to these two kinds of genetic information that I mentioned a moment ago, that you have both LD and relationship contributing to genomic prediction in the BLUP method. How do different design parameters influence the way accuracy, how accurate your predictions could be? So that's what this table attempts to show. The first main thing you can vary is the training population size. A larger training population size will have a very strong influence on the accuracy due to LD. On the other hand, the simulation studies suggest it has a more ambivalent relationship I used the relationship twice. It has the more ambivalent effect or connection between accuracy due to relationship. Meaning, if you, for example, construct a simulation where you only have relationship information and you have no LD, a clever way of doing that is to put all of your markers in QTL on different chromosomes in a simulation, and therefore your markers can't have any LD with the QTL, and yet you still get prediction accuracy. You don't see very strong dependency on population size in those simulations. A corollary of this is that if you're starting off with genomic prediction and you have a relatively small training population, the accuracy you're going to achieve is largely due to the fact that the markers are capturing the relationship information. It takes, in a situation where both LD and relationship contribute, it takes a relatively large population size before LD will start to become more important than relationship. And then the reason that's important to mention is because as you start to apply your predictions and you look at how many generations of recombination are separating your training population from the selection candidates or the validation population, which could, for example, if you tried to do multiple cycles of selection in a greenhouse and you don't have a chance to update your training set, how accurate would the predictions be by the third generation, let's say? Well, it turns out that the accuracy will decline relatively slowly if LD is playing an important role in the accuracy. However, if relationship is playing a more important role, you're going to see a fairly rapid decline in the accuracy of your predictions. So this is where having some understanding of whether LD or relationship is playing a more important role in your situation can help you understand some of the potential limitations of deploying your training population. Some other ways of thinking about this space of predictions that follow from what I've said already. This is a traditional two-way classification of genomic prediction. On the rows, we have are the genotypes that we're trying to predict, are they part of the training population already? Are they not part of the training population? And likewise for the environments. Again, so if I were trying to, let's say, make breeding value predictions for a set of individuals or clones, that I've already collected some phenotype data for, and I'm interested in their breeding values relative to the set of environments that I have collected data. That's the upper left corner. That's relatively easy, not in the sense of whether it's hard to actually get the number to be predicted, but whether the predictions are going to work in practice. It's fairly easy to get them to work because you'll expect high accuracies. As you start to move towards environments that are not represented well in the training population, or individuals that have never been phenotyped before, it gets harder. And the most difficult in terms of achieving a high accuracy would be a set of individuals with respect to a set of environments that you have never measured before. So hopefully that's pretty intuitive based on what I've said up to this point. Here are some figures from some classic papers in the literature that showed some of these phenomena with some real data. This is a publication from 2013 showing in a set of maize double haploid families, we're going to try and predict the progeny of a single biparental cross, A by B. So A and B are represent the names of the inbreds of the parents. And then on the y-axis, we have the prediction accuracy, and then we have the training population size on the x-axis. So the different curves here correspond to different amounts of information or different kinds of training populations with respect to their relationship. So if, for example, I already have some full sibs of the selection candidates, because I've already created and phenotyped some other A by B individuals, that's going to give me the highest accuracy possible relative to the next set of curves, which correspond to training sets in which we don't have any full sibs, but we only have half sibs available, but we have half sibs through both of the parents. A has been crossed with some other inbred C, and B has been crossed with some other inbred D. In both situations, you see the dependency on the training population size, because we are only looking at our selection candidates are only within a population, within a biparental population. They all have the same pedigree relationship. So it's easier to deconvolve the different contributions to the accuracy. But then moving on down this series, if only one set of half sibs is available, you have still lower accuracy. And when you don't have either A or B represented in the parents of the progeny that are being phenotyped, you have the lowest accuracy. So that's a good example from a plant breeding. Here is one of the classic papers from animal breeding. I believe this was a sheep data set, and they were trying to understand how can we rationalize after the fact what accuracies we're observing in their predictions. So the y-axis is the accuracies, and the x-axis is two different measures of relationship. On the left-hand side, they just took for every individual, they took the average G coefficient with all the other individuals in the training set. And you can see from the figure, it doesn't really seem to explain what they're observing for accuracy. Whereas on the right-hand side, they're only taking the average G coefficient for the top 10 closest relatives, meaning the top 10 highest G values in the data set. And that does seem to be a good predictor. So again, showing how close relationships can dominate accuracy, but then being careful about how that may play out as you start to apply them over time across multiple generations. Okay, so that kind of wraps up some of the more theoretical, or I guess the highly condensed version of what I would teach over maybe a couple of weeks to a graduate student course. I'm going to pivot now and talk a bit more about the specific software and some examples of making these predictions. And I thought I could just pause for a second and I'll just peek and see if there's anything pressing in the chat that anyone wants to address at this point? Anything I should look at that would help to clear up something I've said so far? Mohammed asked, in a trait with a bigger dominance variance and additive variance, does the GS strategy work? And I think Margaret gave a good answer there. Yes, I'm not going to actually talk much about dominance variance. I presented what I showed you so far was just based on this concept of splitting additive and non-additive, but we can actually start to then further partition the non-additive into dominance. And of course, in polyploids, dominance has different degrees, and there's epistasis, and you can fit a lot of different covariance models. But again, that would be, I think, beyond what I would aim for in a kind of first course in this. But there is a lot that one can do, and these especially when you want to predict the genotypic values, these modeling non-additive covariance can help with that. But for breeding values, which is the most important thing about genomic selection, is predicting breeding values. The additive is the most important thing to focus on for right now. And Patricia is making a comment about close relatives and being careful about inbreeding, which is a point I'm going to come back to at the end. So yeah, thank you for raising that here, Patricia. Okay, well, that gave me a chance to at least pause my thinking. So let me go back to hiding the controls, and I'll keep going, and we can do some more Q&A later. No, there we go. Okay. All right. So yes, how do you actually do this in practice with the complex data set that you're likely to deal with if you're a plant breeder? And we talked a bit about this yesterday in the context of GWAS. It's a very similar idea for genomic selection. It's common to try and split the analysis into two stages. In stage one, you're going to analyze each of your environments separately, compute a best linear unbiased estimate, or BLUE, for the genotype as a fixed effect, and then use that as the response variable in stage two. And that's where the marker data will be used in stage two to compute the breeding values or the marker effects. And there we go. So I've already mentioned that by using the variance covariance matrix for those stage one BLUEs in stage two, you can do a better job of capturing all of your data. That's called fully efficient. What I want to show you is this package StageWise, which can help with that process. The first vignette covers analysis of a single trait under the assumption of homogeneous G by E, meaning we're going to assume that the genetic correlation between all of our environments is the same, which is a reasonable assumption if, for example, you had a single location in multiple years. Even if you had a set of similar locations, it would still be a reasonably good approximation. As you start to have multiple locations, which are, it's not appropriate to assume that they have the same degree of similarity, you're going to want to model a more heterogeneous G by E, and that's what the second vignette illustrates. And then finally, in vignette 3, we'll talk about correlated traits and how to make predictions for correlated traits. So this is the workflow of the package, and we talked a bit about this yesterday. Stage one is where you analyze each environment separately. Read Geno makes your G matrix. Those get fed into the stage two, which uses ASRMOR to estimate variance components. There's this intermediate step to invert the full variance covariance matrix. This is where you can also mask the BLUEs for specific individuals or individual trait combinations and do some interesting cross-validation when you make the BLUPs. So we're going to focus today on this lower branch, which is the predicting of the breeding values and the genotypic values. So this first example comes from a potato breeding program at the University of Wisconsin with clones from other programs, but all the data came from a single location over 6 years. There's about 1,000 clones, and the marker data comes from a SNP array with about 12,000 after it's actually two different versions of a SNP array that were put together. And I'll say a bit more about that later. So here's the read Geno command. It takes as input a file name. You tell what ploidy. This doesn't yet work for mixed ploidy, but again, that would be something that should be able to implement this year. You have the option of including a map. That's mostly useful if you're using it for GWAS. And then there's some a parameter here that you can use to do some filtering based on minor alleles. The syntax for stage one looks something like this. It just the command also takes as an argument the name of the phenotype file. So the phenotype file has columns for the ID column, it has an environment column, and then it has whatever other effects you may want to model. So in this example, we have used principally complete block or incomplete block design. So for the first 5 years of the data set, which is from 2015 to 2019, we have a column named block. And I've also included a column called stand count, because I'm going to use the stand count to adjust my yield, realizing that the plants can compensate a little bit. And you specify for each of your effects, are they fixed or random, and are they factors or variates? The output from stage one, it's pretty basic. You get an output of the blues on the one hand, you get an output of the heritabilities on a plot basis. So this is just the ratio of the genetic variance to the sum of the genetic and residual variances. You get some basic standard visuals like QQ plots and box plots that can help you eliminate outliers. So it's not, again, meant to be a full service software. There's lots of other good software, and the nice thing is you can use whatever software you like to use, and you can still feed all that into stage two. Stage two is a bit more difficult and to do on your own unless you become very good at running ASRMLR, but it's certainly possible. I mentioned one of the motivations of doing a two-stage analysis is that you may want to fit different models for spatial variation in stage one. And there's this nice R package that was developed by Wageningen University called SPATS, and you can actually use that in stage one. It just calls SPATS and takes the output and puts it in the right format for stage two, and you get some nice visuals of these 2D splines. Fortunately for our potato trials that at our breeding farm, which is irrigated and very flat, we almost never have any real spatial variation to worry about, which is why historically we've used just simple blocking designs, and they work pretty well. Okay, on to the stage two model. What does it look like in more detail? The response variable is the blue from stage one for genotype I in environment J, and that's written as some environment effect plus this additive value for the clone, and the additive value is multivariate normal with the G matrix as the specifying the covariance. At the moment, there is no modeling of covariance structure for non-additive. And again, that's something that is planned for this coming year, but we've been able to do quite a good job, I think, analyzing our data sets just by treating the non-additive as an independent genetic effect. The advantage is that it's more agnostic about what the actual covariance is, and it can also account for misspecification of your additive covariance, right? The G matrix you calculate from your markers is not the true G matrix. So some of that additive value could potentially get estimated as part of the residual genetic value, which is why I tend to call it residual rather than non-additive in this context. The unique part that makes this fully efficient is that there's this random effect that represents the stage one error, but there are no variance parameters estimated. The variance parameters are all fully specified by the output from stage one. And then the residual in a statistical sense in stage two is actually the G by E. So it's called G by E in the output, but in the stage two linear model, it's actually the residual. So after stage two, you run this BLUP prep command, which takes as arguments the blues, the variance covariance matrices, your marker data, and then these variance component estimates from stage two. And then if you run it with this what equals ID, you just get a table, and the table includes the breeding values, the genotypic values, which is the sum of the breeding and this residual component, residual genetic component. And then you get reliabilities for both of those. And I'm a big fan of reliabilities. It just makes, it's very easy to get a sense of trying different ideas if you're a breeder. So one of the, I think the unique things here is that that's built in from the beginning, that you're always going to see that in your output. We had talked a little bit yesterday. There were some questions about, can you include fixed effects in your predictions? And yes, it can be done quite easily just by adding the names of the markers when you call stage two as this fixed effect marker. Then you will see the partitioning of variance, but then when you make your predictions, they also are used in the breeding value predictions. One of the, I think, kind of revolutionary outcomes for plant breeding that genomic selection has enabled is that you can you can approach multi-location testing in new ways, right? Traditionally, you in a traditional breeding program, you don't start to put your clones or your entries in multiple locations until you have enough seed to be able to put to actually plant at least a plot in each one of those locations. You wouldn't put 25% of your germplasm in one location, another 25% in another, something like that. But that kind of a highly unbalanced design actually is quite feasible when you start talking about genomic prediction, because the marker data essentially help you connect these different locations. And we say alleles are replicated even when genotypes are not. So even though there is some bias potentially in your estimates, you can show, many papers have shown this in the last decade, that you over time, this is a much more efficient way of running a breeding program, because you're starting to get estimates of breeding values that sample your target population of environments at an earlier stage than you would otherwise. So even though you don't have enough seed to put the clone everywhere, you start to learn something about its wider adaptation. So that's something just to file away for thinking about how to design your breeding program. How do you actually analyze multi-location data sets? That's what I want to come to now. And that's the topic of vignette 2. So this is it's also a potato data set, but it's a different set of, some subset of the clones that were tested as part of the national trialing set for potato. So there are 9 different states you can see here. 9 states participated in this over a period of 6 years, I think we have, from 2011 to 2016. The main difference in terms of the input file is that you have this additional column that's called LOC for location. Now, you don't have to, there are modeling heterogeneous G by E doesn't have to be location. It could be something else. It could be a management system, for example. But because this is the most common situation where a plant breeder is going to be interested in this, that's just hardwired. If you are using a management system, you have to just call the column location and realize that that's just the keyword that the software uses. Right, when you start to look at, this is spanning the entire United States, basically north to south, east to west, you start to see there are a lot of differences in terms of the plot-based heritability in different research settings. Right, this could be dependent upon the climate of a location, but also just what equipment they might have or what experimental designs they're using. A lot of things can go into this, but we learned something about how much you can trust the data from one plot if you're talking about the data coming from New York versus, say, Florida, for example, in this particular context. So this is a figure that you would get from stage 1. Now coming to stage 2, the way that the heterogeneous covariance is modeled is using the factor analytic capabilities in ASRMLR. So if you are familiar with ASRMLR, you have the possibility of specifying how many latent factors you want to use to try and capture that covariance. My experience is that 2 latent factors is enough complexity to handle most of the practical situations that a breeding program would want, and it just simplifies the way the software, it simplifies the interface and the description of how someone can use the software. If you want to be able to have more control of it, then of course you can just do your own analysis with ASRMLR. But for StageWise, at least, it's constrained at having these 2 latent factors. The caveat that in this vignette data set, it's not really a breeding program. It's 9 breeding, well, it's not 9, but it's 9 locations and it's over 5 breeding programs. So it's maybe not surprising that we're not going to be able to fully explain all of the covariance with only 2 latent factors. But anyways, here you can see how when you run this type of analysis with stage 2, when you run this summary command, you get, in addition to the proportion of variance explained table, you also get this matrix of the genetic correlations between the locations. You also see that the partitioning of variance looks a bit different here. You have this G by location showing up because that's a typical way we represent that. Even though it's not a compound symmetric model, you can calculate on average what the G by location variance looks like. And notice also that this example doesn't use marker data. So that's why there's no partitioning of the additive and the non-additive in this situation. This is a large data set. If you tried to add marker data with all 9 locations, it would take quite a while. So I recommend, might as well just try and get a sense for what your structure of your covariance looks like, and then you can maybe hone in on what set of locations you're more interested in. So for example, this is what the visual representation of that 9x9 matrix. One convenient way of representing that is with what's called a uniplot, a very clever technique developed by Cullis et al. A decade ago. And it shows graphically on this unit circle two types of information. The distance of the point from the center of the circle, that squared radius, is the proportion of the variance for that location explained by the latent factors, meaning that locations that are on the unit circle are perfectly explained by your two latent factors, and those that are not, you probably aren't really doing a good job of representing their correlation with the other location. So in this example, Texas and Florida stand out as being quite a bit different than the other locations. We're not doing a good job of representing them. For locations that are on the units on that red circle, the separation, the angle that's subtended, if you think of a piece of a pie or a wedge, the larger that angle, the lower the correlation. So it's the exact relationship is the cosine of the angle is equal to the correlation. The cosine of 0 is 1, which is why when they're closer together, they're more related. So what I did is I focused on, I'm the potato breeder for Wisconsin. I'm going to focus on a set of locations that were more similar to Wisconsin and also were well explained by the two latent factors. So I've reduced it down to this set of 6 locations. Now I've added the marker data. And now when I run the summary command, now I do see the partitioning of the genetic variants into the additive and the residual genetic components. And furthermore, when I look at the correlation matrix output, the numbers above and below the diagonal are no longer the same. The numbers above the diagonal are the additive correlation, and those below the diagonal would be the total genotypic correlation. Oh, and just to look at this figure and see what does it show. So it shows that the Missouri, Oregon, New York, and Michigan, they're quite similar to Wisconsin in terms of genetic correlation. North Carolina, although it is explained well by the data, it's just not as highly correlated. So this brings me to the first illustration of what I think is one of the very attractive features of the software is constructing selection indices, and the software makes it very easy to use the optimal index because it has a multivariate BLUP capabilities. And under multivariate BLUP, you have the nice property that whatever linear combination of breeding values you want to specify as the true genetic merit, that's what this first line says. Some breeding values, this is a sum over, in this case, locations. So I'm going to maybe specify some weights for different locations. Maybe Wisconsin has the most weight if I'm the Wisconsin breeder, but I also want to perhaps have varieties that can do well in other locations, and maybe they get a little bit less weight. But whatever that weighting is, the index is supposed to be the best predictor of that merit, and those weights are now the same coefficients of the BLUPs that you would calculate, provided the BLUP calculation is done in a multivariate way. Now, the weights that you specify have to be standardized to be sensible, especially when you start talking about combining different traits where it's an apples and oranges thing. So there is a standardization that is applied, which you can think a little bit more about if you start to do this in practice. So in this application, we're going to compare the reliability of the breeding value predictions for Wisconsin versus North Carolina. And based on the observation I just made that these 4 other locations are more similar to Wisconsin than North Carolina, I would expect that I can borrow information from those correlated locations and achieve a higher prediction accuracy or a higher reliability. And that is in fact the case. This figure on the right shows the reliability of breeding values for Wisconsin above on the y-axis, and the x-axis is the breeding values for North Carolina, and all the points are above the dashed 1-to-1 line. The syntax in the software is pretty easy to do this. You just specify some vector, which has a named vector. So you give the names of the different locations and you put the weights. So the Wisconsin index has a weight of 1 for Wisconsin and 0 everywhere else. And similarly for North Carolina. And then when you call the BLUP command, there's this additional argument that's called index weights. And that will then compute whatever combination of breeding values you're interested in. So I don't have my time in front of me, but hopefully I'm about right in terms of timing. And this is kind of a good transition point to talking about Vignette 3, which I thought we would do after our break. And I'll just stop by thanking the funding and also all my colleagues in the potato industry who have helped to collect this data over a long period of time.